Welcome to The Explainer. I am so incredibly pumped to have you here today because we're about to dive into a literal biological mystery. We're going to be unpacking some incredible new research that completely shatters how we perceive nature's illusions. We always hear that seeing is believing, right? Well, today we're going to learn exactly why, in the natural world, our own eyes can completely and totally deceive us.
Think about this for a second. If you walk into a forest and you see two trees that look entirely identical, the exact same height, same exact leaf shape, the bark is peeling in the exact same way, are they the same tree? Logic says yes, absolutely. But nature? It turns out nature is the ultimate catfish. We're going on a journey today into a secret world that's been hiding right in plain sight.
Section 1, the Ecozone Crossroads. To actually solve this mystery, we need to travel to a very specific, super dynamic part of the world, where totally different ecosystems just crash right into one another.
Okay, let's dive into this. Looking at this map of southwest Nigeria, you can clearly see two distinct zones. We've got the dense, humid lowland rainforest shaded in green, and right next door, the drier, much more open derived savanna shaded in yellow. Now, right across this fragmented, constantly transitioning landscape grows a really vital canopy tree called Pterocarpus santalinoides. It's a massive lifeline for local communities there, providing everything from timber to traditional medicine. But here's the catch. This landscape is changing super fast due to deforestation and climate shifts, and scientists desperately need to understand exactly how this tree is managing to survive across both zones.
Section 2. The Illusion of Sameness. So, to figure out how to protect these trees across such vastly different environments, researchers took the most logical first step: a massive visual field test.
The scientists went out and meticulously analyzed the physical traits of these trees out in the wild. And they didn't just glance at a couple of things either. They documented 30 different vegetative traits. We're talking leaf architecture, like having an ovate shape or an entire margin. Leaf surface traits, like a smooth texture or a shining surface. And even stem and bark characteristics, like having a smooth stalk and gray-striped bark. Going into this, they completely expected that a tree baking in the dry savanna would look fundamentally different from one soaking up the wet rainforest. It just makes sense, right?
But here is the crazy part. After analyzing 19 different samples from 5 wildly different locations across the rainforest and the savanna, the physical traits showed absolute uniformity, 100% phenotypic stasis. What that means is their physical form remained completely unchanged, regardless of where they lived. Every single tree looked exactly the same. The researchers were literally staring at a perfect physical illusion. Based entirely on what their eyes were telling them, these trees were completely identical.
Section 3. Tracking the maternal lineage. Thankfully, the researchers didn't just stop at what they could see. They decided to peel back this physical disguise and dive straight into the genetic reality. They started by looking at a very specific type of genetics called chloroplast DNA, or CPSSR for short. Let me break this down simply for you. In these types of trees, this specific DNA is passed down strictly from the mother. Think of it like tracking the mother side of your own family tree, but, for a plant. This DNA is carried inside the heavy seeds of the tree, and because those seeds are so heavy, they usually just drop straight down, really close to the mother tree. They don't travel very far at all. So this gives us a practically perfect map of the tree's historical maternal family tree.
And boom, here is the massive plot twist. When the scientists actually looked at this seed DNA, they didn't find one identical population. No way! They found 3 completely distinct maternal lineages: haplotype Th4, haplotype Th5, and haplotype Th6. That is 3 entirely different evolutionary histories, all hiding behind one identical physical disguise.
Now, what's really interesting about this slide is just how strict this geographic partitioning actually is. Look at the pie charts here. The maternal lineages are completely isolated across the map. The Th4 lineage, completely isolated down in the Iki rainforest. The Th5 lineage, totally alone over in the UI savanna. The seeds are just dropping straight down into the dirt, creating these isolated pockets of totally unique genetic history that never ever mix, even though, again, the trees look exactly the same.
Section 4. The pollen connection. At this point, you might be thinking, okay, mystery solved. But wait, because we have a massive biological contradiction coming right our way.
To get the full picture, the researchers knew they couldn't just look at the heavy seeds. They also had to look at the nuclear DNA. And here is the absolutely vital biological difference between the two.
While seed DNA, that maternal line, stays super close to home. Nuclear DNA captures inheritance from both parents. And how exactly does that spread around? Through pollen. Pollen is light. It gets carried across vast distances by the wind and by flying insects. When they mapped out this nuclear, pollen-driven DNA, they got this fascinating similarity matrix. Notice those deep blue squares? Those represent incredible genetic similarity. We're talking up to 98.6%. So, completely contrary to the deeply isolated maternal seeds we just looked at, the nuclear DNA reveals that these widely separated rainforest and savanna locations like Oahu, Fren, Ui, and Olakamegi are highly connected. Their nuclear DNA is almost entirely homogenized. They are basically constantly talking to each other genetically.
And this dendrogram brilliantly illustrates that exact contradiction. Notice how tightly most of these populations cluster together on the right side? They're constantly sharing genetics via pollen. But then look at the very top line. The isolated Icarae lineage stands completely alone, distinctly separated from the rest of the entire group.
Section 5. Asymmetric gene flow. So how in the world do we reconcile this? We've got a tree that looks perfectly identical everywhere, has deeply isolated maternal seeds, and yet shares highly connected nuclear DNA all across the landscape.
Okay, it all boils down to this number right here, a discordance score of 0.73. This score, which is 0.73 out of a maximum of 1.0 by the way, mathematically proves a massive decoupling. It represents a phenomenon scientists call asymmetric gene flow. Basically, the way the tree looks, the way its seeds drop, and the way its pollen spreads are all operating under entirely different biological rules. Let's walk through this mechanism step by step just to really lock it in. Step 1. Specialized pollinators are carrying pollen across vast distances, effectively homogenizing the nuclear DNA across both the rainforest and the savanna. Step 2. At the exact same time, the tree produces heavy seeds that drop right next to the mother trunk, strictly isolating the maternal DNA in specific locations. Step 3. This pollen-rich seed-pore dynamic ends up creating a hidden evolutionary map. Deeply unique genetic histories are literally hiding beneath a totally uniform, identical physical cover.
Section 6. Rethinking conservation strategies. Now, this isn't just some cool academic puzzle. Solving this genetic mystery is absolutely crucial for the real-world survival of these West African ecosystems. Think about it. If we only used our eyes to guide conservation, we'd probably assume saving any one of these trees would be enough, since they all look exactly the same, right? But the genetics tell us a totally different story. Thanks to this data, we have highly actionable takeaways. First, we absolutely must protect what we call evolutionary hubs, like Olakamegi. It has a massive haplotype diversity score of 0.733, simply because it acts as a vital mixing zone where different maternal lineages meet up. At the same time, we have to preserve isolated lineages, like Aikiri, which is a 100% pure historical survivor of that Th4 lineage. If we lose Aikiri, we literally lose an entire irreplaceable branch of evolutionary history.
The source material summarizes our key takeaway perfectly with this quote: "Conservation frameworks must transcend phenotypic assessments to prioritize these evolutionary significant units. Simply put, if we want to save our forests from climate change, rapid human expansion, and deforestation, we can absolutely no longer judge a forest purely by its cover."
Which leaves us with a truly fascinating question to end our explainer today. If this one West African canopy tree managed to hide 3 completely different evolutionary lineages behind an identical physical disguise, well, how many other identical-looking species out there are hiding secret evolutionary histories just waiting to be saved? Nature is infinitely more complex than it appears on the surface, and honestly, our exploration is just beginning. Thank you so much for joining me on this incredible deep dive today.